In this video, I'm going to discuss two questions on Chapter 8, Recombinant DNA Technology. The first question here is about the artificial plasmid called PUC18. Always read the question. Figure 1 shows the structure of an artificial plasmid PUC18. Let's learn the structure of the artificial plasmid here. This plasmid, it consists of the LAC-Z gene, and also within the LAC-Z gene there is this part where we call the multiple cloning site. What is a multiple cloning site? It consists of many restriction sites, which will be recognized by different restriction enzymes to cut the plasmid. Another part is the ampicillin resistance gene, the antibiotic resistance gene. This is where it acts as the selectable marker gene. These two parts are very important for the plasmid to be used in recombinant DNA technology. The first question here, it asks you to state the role of PUC18. The role of plasmid, of course, is to act as the cloning vector. And after that, you have to give two of its characteristics. Make sure you answer based on the question. You must first answer its role, which is acts as cloning vector, and then only you give the characteristics of the plasmid. Give any two from these three characteristics of plasmid. Being a cloning vector, the plasmid must be able to accept foreign DNA in the multiple cloning sites, which is the part where I mentioned just now. This is the part where the restriction enzyme cut. The plasmid will also be able to replicate freely in the host cell, as well as it has the selectable marker gene or genetic marker, which in this case the ampicillin resistance gene. The second question here, based on figure 1, what would happen to the PUC18 if EcoR1 is used? If you know that EcoR1, it is a type of restriction enzyme. Restriction enzyme will recognize the restriction site and then cut at the restriction site. So in this case, the restriction site is at the multiple cloning site. So what would happen to this plasmid if the restriction enzyme EcoR1 is used? We say that the plasmid or the PUC18, it will be cut by the EcoR1 enzyme at this restriction sites. Make sure you give the sequence of the restriction site recognized by EcoR1, which is 5' GAA TTC 3'. State the direction of the sequence. And what would happen after the plasmid is cut? It will produce compatible, or we say complementary, sticky ends. EcoR1 is one of the type of restriction enzyme that will produce sticky ends, while another type of restriction enzyme that you learn, the SMA one, instead of producing sticky ends, it will produce blunt ends. C. According to your answer in B, explain what would happen if no foreign DNA is inserted into the digested PUC18. Digested, in this case, it means open. After this plasmid being cut open, you have to answer this first part, and then only you answer the second part, which is about its effect on the blue-white screening. So what would happen to the plasmid if there's no foreign DNA inserted at the multiple cloning site? If nothing is inserted into the open plasmid, then this plasmid will then join back together. That means the LAGZAC gene, it will be intact. It will not break open. So we say the sticky ends of the plasmid will join together by the enzyme DNA ligase. The plasmid simply reclose on itself. How would this affect the blue-white screening? Because the plasmid rejoin itself, the LAGZAC gene becomes functional again. And that's why beta-galactosidase can be synthesized normally to hydrolyze the Asexual. This will make the bacteria that took up this plasmid which is rejoined and reclose itself, having the functional LAGZAC gene, will form a blue colonies. And the last question here. Why is it important for PUC18 to have an antibiotic resistance gene? Why must a plasmid have this antibiotic resistance gene? Having the antibiotic resistance gene, it helps to identify or select the bacteria that have taken up the plasmid. It helps the scientists to identify which bacteria have taken up plasmid and which bacteria don't. And only bacteria that have taken up the plasmid will be able to grow. This is the use of the antibiotic resistance gene, which makes it easier to identify the bacteria with the plasmid. And please remember that when you write LAGZAC gene, there is no spacing and you have to underline all. While equal R1 is also the same, there's no spacing and you must underline equal. And please remember 1, you must write Roman number 1. Same goes to small 1, no spacing and Roman number 1. And you have to underline small. The second question here is about using cDNA to produce insulin. So the question say, Figure 2 shows how insulin can be produced using genetic engineering, which is this diagram here. You can see how the plasmid is isolated from the bacterial cell, as well as a DNA, the target DNA, is isolated from the donor cell. After that, through insertion, the target DNA is inserted into the plasmid and then transformed into bacterial Y. Inside bacteria Y, the gene will be expressed to produce the protein, the hormone insulin. And this bacteria which can produce insulin will be cultured in a fermenter, a container where the bacteria will reproduce a lot to produce insulin. So, the first question here it asks, the target DNA in figure 2, which is this one here, is cDNA. Describe briefly how insulin cDNA is formed. cDNA is the type of DNA that contains only exons. How to produce a DNA with only exons? First, you must get the mRNA. In this case, we get the mRNA for insulin from the human pancreatic cell. And this mRNA will then be used as a template to produce the first strand of cDNA by using enzyme reverse transcriptase. You must mention the template, which in this case the mRNA, as well as the type of enzyme used, which is enzyme reverse transcriptase. These two, they are needed to produce the first strand of cDNA. DNA is double-stranded. That's why we will need to produce another strand to make this cDNA double-stranded. So what do we need to use to produce the second strand of cDNA? we need the first strand, the single-strand cDNA as template, as well as the enzyme DNA polymerase. These two, they are needed to produce the second strand of cDNA, and that's how we get a double-stranded cDNA. The second question asks, if the target DNA in figure 2 is not cDNA but nuclear DNA, the genomic DNA, original DNA, why can't the bacteria express the insulin gene if nuclear DNA is used? Explain this statement. Nuclear DNA is the type of DNA that consists of both introns and exons. If you still remember what you learned in chapter 6 about RNA splicing, then you should remember in prokaryotes or bacteria, they do not have RNA splicing. Absence of RNA splicing mechanism in prokaryote or bacteria. That means bacteria cannot remove the introns from the DNA because they simply do not have the mechanism to remove the introns. This will cause no functional protein could be made, or we say it produces non-functional protein. That's why if a bacteria is used as a host, then the target DNA used, it must be a cDNA. The third question suggests a method used in genetic engineering to detect the bacteria Y. This is the first part of the question, and then you have to describe this method briefly. How can we detect which bacteria have the recombinant DNA that can make insulin? We use a technique called blue-white screening. Blue-white screening allows us to identify the bacteria with plasmid or without plasmid, as well as the bacteria with recombinant plasmid or non-recombinant plasmid. The one that we want here, bacteria Y, is the bacteria with recombinant plasmid, which means the plasmid with the insulin gene. What would happen to bacteria Y during blue-white screening? Bacteria Y, which consists of the insulin gene, it will form white colonies. Why? Because the LAGZAC gene in the plasmid is non-functional. That's why it does not produce the beta-galactosidase enzyme and thus unable to hydrolyze Asexual. This causes the bacteria Y to form white colonies. And the last question here. The bacteria in figure 2 reproduce asexually in the fermenter. Explain the advantages of asexual reproduction for insulin production. Why is it good to have asexual reproduction in genetic engineering? Simply because asexual reproduction occurs rapidly, so it can produce insulin rapidly. Insulin can be produced in large amounts, as well as when the bacteria reproduce asexually, all the bacteria in that culture, they are genetically identical, which means they have the same insulin gene and they will produce the same insulin protein.